Here we are at day 5. Let's review what we have done so far. We took the samples you collected and broke them into 3 kilobase segments. We inserted those segments into plasmids. We inserted the plasmids into E. coli bacteria. We used the bacteria to both duplicate the plasmids and to sort the fragments of DNA that we want to sequence. We then placed each bacterial colony containing a discrete segment of sheared DNA into a separate well of these 384 well plates. We then broke open the E. coli bacteria to release the plasmids. We replicated the plasmids using rolling circle amplification. Next, we added fluorescently tagged nucleotide, Taq polymerase, untagged nucleotides, and buffer to the amplified plasmids containing the inserted DNA segments to create Sanger fragments through thermal cycling. Finally, we washed away all the non-DNA material. At this stage, we are actually ready to start reading the sequence of the genetic material. Here we will load the 384 well plate into this ABI 3730 sequencing instrument. Inside this sequencing instrument sits this unit, the capillary array. This array has small glass tubes that are filled with a polymer matrix. This matrix has holes that allows the DNA to migrate through, and based on the fact that DNA has a negative backbone, it will migrate toward the positive anode region. The smaller fragments of DNA will migrate through faster than the larger fragments. As they come across this detection cell, there's a laser that shoots through both the top and the bottom, and it excites the dye terminus on the end of each fragment. This dye terminus then emits a light code back to a CCD camera, and it presents this image that you see here on the screen. This is each of the capillaries, 1 through 96, and each well is represented by one of these single reads going across the screen. If we look at the screen, we can see the sequence as it is being read. This is a major accomplishment right here. However, we are far from finished. All this raw data needs to be assembled back into the genome of the organism we are sequencing. To do this, we send the data to our assembly team.